So this exam was not entirely... we are moving on then into Chapter 5. There's two different topics that this chapter covers. It sort of squishes them together. They have a little bit to do with each other, but not a huge amount. We're looking at energy and the cell, so we're going to look at sort of how cells use energy and what kinds of energy they use. And we'll also be looking at some particulars about the cell membrane. As I said, since in Chapter 4 we've sort of done a whirlwind tour of the cell, the next job to do is to look at some of these organelles in detail and ask, what is their vital function for the cell? How do they achieve that vital function? That's why I say that the next exam is going to be a fairly difficult one, because we're going to be looking at an awful lot of energetics, enzymatic reactions, and a lot of biochemical pathways that at first glance may seem very complicated. What I'm going to try to do is make them as simple as possible and reduce the amount of stuff that you have to memorize so that you don't have to worry quite so much about those reactions. The reason that the cell does them is far more important than remembering, you know, step A goes to step B goes to step C. Those things are less important. Okay, so some of the major points that were presented in this chapter. For part 1, energy in the cell, one of the more important points that's presented is that cells do in fact follow the laws of thermodynamics. You will find certain spiritual groups who will vehemently claim that cells do not follow the laws of thermodynamics, and therefore there must be an outside mystical energy source that gives them their energy. All right, well, I'll demonstrate fairly clearly that that is simply not so. Thermodynamics does in fact apply to living systems. Okay? Chemical reactions store or release energy. You can actually store energy in a particular chemical form, in a chemical bond. You can release that energy to do some sort of work for the cell. Right? And we do that all the time. We store and release energy. Right? Energy can be stored in lots of different forms by cells. It's not only one form that's the sort of universal energy equivalent of the cell. There's many different forms that the cell uses. Okay? And another point is that cells have to be able to do chemical reactions effectively, efficiently, and without a lot of excess heat being produced or excess energy being lost. And so they use enzymes. Enzymes are proteins or sometimes even RNA molecules that can do chemical reactions. Okay, for part 2, the cell membrane, we're going to find out the cell membrane is a structure called a lipid bilayer. In fact, I've called it that a few times, sort of accidentally, because it's sort of hard for me to hold back from that terminology. I'm so used to it. But the cell membrane is, in fact, a lipid bilayer. That means there's 2 layers of lipid molecules that are contributing to formation of the cell membrane, and indeed all of the internal membranes in a cell as well. The membranes around the mitochondrion are lipid bilayers. The membranes around the rough ER, the smooth ER, that's all lipid bilayer. Okay? Embedded in the cell membrane are proteins that provide a wide variety of functions. They transport molecules in and out of the cell. They may allow signals to be received by the cell. They may increase the structural strength and integrity of the cell. All of those things are possible. Okay? And then we'll talk a lot about diffusion. Diffusion really strongly affects how cells use their cell membrane. Diffusion is the movement of substances until they reach an equilibrium, as I'll show you. And because water tends to go where those solutes go, it's critical for cells to be able to control how much solute they've got in them and regulate that with respect to what's outside. Otherwise, they're going to gain too much water or lose too much water, and they're not going to survive. Okay, so lots of important points in this chapter. All right, first of all, what is energy? Well, if you want a good definition of energy, you better take physics, because you're not going to get it here. This is my sort of off-the-cuff definition, and it's very similar to the books. It's the capacity to perform work, okay? To move or change matter. And you're doing this in a way that the matter wouldn't change if you just left it alone. Right? You're adding something into it to change that matter in some way. Right? That's energy. There's two major types of energy we can talk about. Kinetic energy, which is energy being released. Okay? It's actually doing work. And then potential energy, which is stored energy. Right? In cells, we deal a lot with potential energy. There's a lot of energy storage that goes on in cells in order to allow them to do chemical reactions when they need to. Okay? So potential energy is stored energy. If that energy was released, then you could use it to do some sort of work, to change matter in some way. All right, this is a neat little slide that shows potential energy and kinetic energy. The potential energy idea is like this cheetah that's standing still, but it's got all this energy stored up, ready to do work if the need arises. And then the kinetic energy portion of that is shown in the top figure with the cheetah just running flat out. How fast can they run? 60 miles an hour for short bursts? It's pretty amazing. Okay? So it's releasing lots of stored energy. It's also generating a lot of heat because that energy release is not efficient. Right? Energy usage and release in cells is more efficient than a lot of energy usages that we have. Cells get around 30 to 40% of the energy out of a particular molecule, whereas, say, a gasoline engine barely manages 20%. Most of the rest is lost as heat. All right, here's another figure showing both kinetic and potential energy. All right, we've got a dam that's holding water back and then a spillway that's allowing some of that water to be released. The released water is releasing kinetic energy. Okay? The potential energy is all on the backside of this dam. That water, in a sense, wants to run downhill, but it's being held back. So you're generating pressure against that, water pressure against that dam. So what you do is, with these spillways, you're releasing a small amount of energy at a time. Cells do the same thing. If they released all of the energy in a chemical reaction, they would burn up because of all the heat that was produced. So what they do is they release the energy a little bit at a time, much as you do here when you release water through a spillway. Okay, you're releasing a little bit of the energy at a time so that it doesn't overwhelm the system. This is a situation that we call a gradient. All right? A gradient is a situation where you have more of a substance on one side of a barrier than the other. Okay? More stuff on one side of a barrier than the other. So the barrier here obviously is the dam. We've got more water on the back side of the dam, less water on the front side, and so we have a gradient. Gradients are stored energy. And that's something that's extremely important to remember and realize as we go through the next several chapters. Because cells make gradients as a form of stored energy, and then they'll release that energy to do work. Okay? So the water that's flowing through the spillway is releasing the potential energy as kinetic energy a little bit at a time. All right, so what kinds of energy are used by cells? Mechanical energy. That's the energy of movement. Every time you move your muscles, you're converting chemical energy to mechanical energy. The energy of molecules is chemical energy. Energy stored in chemical bonds is really what we're talking about here. Okay? When you break chemical bonds, you can release potential energy as kinetic energy. You can capture that energy, or you can simply let it be released as heat. Right? This is one way that we keep our bodies warm at a constant temperature. We're releasing a lot of energy as heat. Okay? Electromagnetic energy is used. Energy of the electromagnetic spectrum. We're talking about light, mostly visible light. That in the electromagnetic spectrum includes things like infrared radiation, ultraviolet radiation, microwaves, radio waves, gamma rays. So all of those are electromagnetic radiation. We'll talk more about this when we get into the chapter on photosynthesis, chapter 7. But plants and photosynthetic protists use light, electromagnetic energy. Okay, gradients. Just to re-emphasize this point, gradients are stored energy. So a cell can pump molecules across a membrane and create a situation where there's more stuff on one side than the other, and that's stored energy. Then the cell can let that stuff flow through the membrane and release that energy. Right? So gradients are forms of stored energy. Cells also use high-energy electrons. Keep in mind that when we before the first exam, in chapter 2, I guess it was, when I talked about electrons being rotating around the nuclei of atoms in specific orbitals, in specific shells. Those are energy levels. They're associated with a level or an amount of energy associated with that electron. Okay? So cells can actually snatch away those electrons and capture that energy in those electrons and release it a little bit at a time. All right, so mechanical, chemical, electromagnetic, gradients, and high-energy electrons are the major forms of energy that are used by cells. Okay, there are 2 laws of thermodynamics that we're going to deal with. There's actually 3 laws, but we're only concerned with the first 2 as far as our living systems go. All right? The first law of thermodynamics says that in a closed system, and this is very important, closed system, there's nothing coming in, there's nothing going out. In such a system, the amount of energy and matter is constant. You never create or destroy matter. You never create or destroy new energy. You can interconvert. Okay? Einstein showed us that. The atomic bomb shows us that. Right? Every time we blow up a stick of dynamite, it shows us that. But energy can't be created or destroyed. Right? So you can convert matter to energy, you can convert energy to matter, but you cannot create matter or energy in a closed system. Right? In an open system, however, energy can come in from outside, right? So the amount of energy doesn't have to be constant. All right, here's an example. Gasoline broken down by combustion when you're driving a gasoline engine and it releases energy. Some of that is used to power the vehicle. It's 20% or less. It's really inefficient. Gasoline engines are very inefficient. There are better ways of using energy, but this is one that we're familiar with. Okay. Almost all the rest is lost as heat. Right? You haven't lost, in a sense, that energy. I should have put this in quotation marks. You haven't really lost that energy. It's still there. It's just that it's been converted to heat energy. Okay. Or it remains unused in the exhaust molecules. So you're throwing out that energy in the exhaust. If the left-hand side and the right-hand side of the equations would be the same in this case, okay, in terms of the amount of energy and matter that you've got. And that's the point of the first law of thermodynamics. All right, the second law of thermodynamics. The total entropy, that is the disorder in a system, in a closed system again, increases. Okay? That's a given. If there's nothing coming in from outside, there's no energy, there's no matter coming in from outside, the entropy or disorder of a system increases. Right? So entropy defined is just the amount of disorder in a system. We measure disorder in terms of things like movement of molecules. You do a chemical reaction and molecules move faster. That's entropy. That's disorder that's been added into the system. All right? The argument that's been thrown out here is that since cells are very ordered structures, they must be violating the second law of thermodynamics. Because if disorder has to increase, then how is it that they can be such ordered structures? Well, it's because they're taking in energy from outside. Yes?
Why is the disorder so important in that dichotomy, I guess? Like, why would you level the amount of disorder in that system?
Why would you level, did you say?
Yeah, why would you level and keep track of the amount of that disorder?
Oh, okay. That's a good question. Why would you keep track of that amount of disorder? It's because that allows you to gauge the amount of energy that's been used in a reaction to do a particular function and the amount that's been lost as heat. Okay, we can say, for example, that a gasoline engine is about 20% efficient because we know that about 20% of the energy actually goes to do work and the rest of it is lost as heat or in bonds that are still in the exhaust molecules. Okay, so that's why we would care about that. We also care about it because to maintain order, you have to fight that disorder. And to do that, you have to use energy. Okay? So an ordered system has to generate greater disorder around it, and that's true of living systems. All right, so here's an example. A cell makes a protein. Protein is an ordered structure. All right, it used energy to do that. But in the process, what it's done is it's lost some of that energy as heat, and the heat made the water molecules around the protein move faster. That faster movement is one of the expressions of entropy. It's greater disorder in the system. Okay? They're more jumbled, they're more chaotic. That's why it's disorder. So it generates greater disorder. Okay? So first law says that the amount of energy in matter is constant. The second law says that order goes to disorder in a closed system. Very important point here that a lot of people forget. This is why they misinterpret the first and second laws. This is only true for closed systems. Right? Here, this individual that's driving the car as well as the car itself are not closed systems. They're receiving energy input from outside. That's why cells can maintain order, because they take in energy from outside. All right? The Earth is not a closed system. We get gargantuan amounts of energy from the sun. And that energy is captured by plants and photosynthetic protists. Right? And it's used by animals and fungi, okay, and animal-like protists. Is the universe closed? We don't know. Okay? There is some, if you're curious, there's some intriguing evidence that suggests that our universe is neither open nor closed, but exactly flat. That may not mean much to you, but we don't really know if there's energy coming in from outside into our universe from outside. So our universe itself may not be a closed system on the whole. Right? The general consensus among astrophysicists and astronomers is that it is. It may not be. Right? The major point here is that the Earth is not a closed system. We get energy from the sun. And that's why cells and organisms can maintain order, because they use that energy. All right. So let's take a quick quiz. And which of the following statements best represents the second law of thermodynamics? When you're thinking second law, always think order to disorder. That's the basic hallmark of the second law. And disorder can be, one way of measuring it is by the rate at which molecules are moving and bumping against each other. All right? So the only statement up there that directly refers to an increase in the disorder of the system is C. Hot exhaust from a car causes air molecules to move faster. That's an increase in the entropy of the system. All right? The rest of these are all various expressions of potential or kinetic energy, but they don't refer to increase in disorder. All right? Okay, so we've got sort of an intuitive idea of what energy is, even though it's sort of not easy to describe it. What we can do now is start looking at chemical reactions and how cells do these chemical reactions. They come in two sort of flavors, depending on whether energy is going in or out. Right? An endergonic reaction is an energy-requiring reaction. Energy has to go into the reactants to make the products. Okay? And the products will have higher potential energy than the reactants. So an energy input is required. Endergonic reactions, you can sort of demonstrate these endergonic reactions because they'll sort of absorb energy from the system around them. So they become colder. Exergonic reactions, however, become hotter because they're energy-releasing reactions. Take sodium hydroxide and dump it in water, and the water heats up. It's an exergonic reaction. All right, those release potential chemical energy. So the reactants have a higher energy than the products. And the difference in energy is what's been released by that reaction. Okay, so endergonic and exergonic reactions. A couple of figures will probably help to see this. This is an endergonic reaction. What we're seeing here is that the reactants have less potential energy than the products. We're just measuring the potential energy of the molecules on the y-axis here. Okay, don't worry about the units or how we're measuring that. We can do that. All right, so the products have higher energy than the reactants, and this is the amount of energy input, the difference between the two. Right? So energy is input, and some of it is stored as potential energy. You could take these products now and react to them to release that energy somehow. Right? Perhaps in an exergonic reaction. So the reactants in an exergonic reaction have higher energy than the products, and so the difference in energy between the two is the amount of energy that's been released by the reaction. Okay? Some of that energy may have been converted to other forms of chemical energy. Some of it may have been released as heat. Okay? Maybe it was used as mechanical energy. All right, so what cells do is they have a lot of energy-requiring reactions. Every time they do a dehydration reaction, it typically requires energy input. Right? How do they achieve that? They do it by doing an exergonic reaction just before the endergonic one. Okay? So they couple these reactions. They use an exergonic reaction and the amount of energy that can be released from that to drive an endergonic reaction. Right? It's, in a sense, what they do is they sort of release this energy. It's stored as potential energy for the endergonic reaction to use. Okay? So coupling of reactions is a sort of a critical concept. Cells do this all the time. All right, the standard energy storage molecule is ATP. To a lesser extent, GTP, TTP, and CTP. You've seen all these before. They're nucleotides. Okay? They're actually RNA nucleotides. Okay? But mostly we use ATP. ATP is a neat molecule because it's got these phosphate bonds over on the left side of this molecule. There's that nitrogenous base, adenine, the sugar ribose. I mean, this looks like a classic nucleotide, except that it's got 3 phosphates. The phosphate bonds are shown as squiggly lines. That means they're high energy. They're unstable. They're easy to break. And when they do break, they release stored energy. Okay? They release potential energy as kinetic energy. Right? So that's what's being shown. The hydrolysis of ATP to break off a phosphate releases a certain amount of energy. Okay. The first two bonds here are the ones that are high energy. So you can break this first bond here. You can break the second bond and get a little bit less energy out. That third bond is really not very energetic. Cells don't use it much. They don't break off 3 phosphates. But they'll often break off 2. Right? And even more often, they break off 1 because that's the highest energy that can be released. All right, so ATP is used a lot to transfer energy to inorganic reactions because this reaction, the hydrolysis of ATP, is exergonic. It's energy releasing. Okay, so you can take the energy inherent in that bond and you can transfer it to another molecule for an inorganic reaction. All right, here in this figure we see the use of ATP to power a particular biological system. This protein is a protein that can move. It's got this little hinge, okay, that can move back and forth, but it won't do it on its own. So what the cell does is it adds energy to the system. It takes a phosphate from ATP, breaks it off, producing ADP, sticks it onto this protein. Okay, so that protein now has increased in energy. This is a coupled reaction. The exergonic reaction of breaking off the phosphate is then coupled to the inorganic reaction of adding that phosphate energy to the protein. Okay, so there's an energy transfer by phosphorylation. Then that energy is released by breaking that phosphate off, and that energy that's released is used by this protein to do a movement, okay, to slide that head back into its original position. Okay, this looks very much like a protein called myosin, which is in your muscles, and it's what allows your muscles to move. Right, so we've got energy release here as mechanical energy. Right? There's a couple of reasons why ATP would be added to this system. One of those is that it allows the protein to move. It adds enough energy to the system to get the protein to do whatever it's supposed to do. But there's another important reason that your book doesn't talk about why ATP is used. It makes the process irreversible. When you add energy to this system, right, and that phosphate is broken off and that protein moves, it's not going to go backwards because that means you would have to add a large amount of energy back into the protein to get it to go backwards. All right, so important point. Cells use ATP to make reactions irreversible. Right? It would really stink if you were trying to contract a muscle and it went backwards on you. You don't want that. Right? So by using ATP, you prevent that from happening. Okay? So cells have to do inorganic reactions to assemble macromolecules, to change the form of molecules, to do a lot of other things in the cell, mechanical movements, that sort of thing, assembly functions. And ATP supplies energy for those reactions. That's where the energy comes from. So ultimately, what we're going to be concerned with in chapters 6 and 7 is how do we make ATP? That's the ultimate goal for the cell, to make enough ATP to store enough energy that it can do all of its chemical reactions. Okay? So if you keep that in mind as we go through those chapters, when we get to them, it's going to make things a lot easier. The ultimate goal is to make ATP, so it can do all of the other things that it needs to do. All right. There's another problem. Are there any questions so far? Makes sense? Yes? What's the difference between ATP and ADP? The presence of that third phosphate. ATP is adenosine triphosphate. ADP is adenosine diphosphate. So ADP is the form in which the phosphate is broken off. ATP is the form where all three phosphates are present. Yeah. Any other questions? Okay, good. There's another problem that cells have to face, and that is the energy of activation. Even if a reaction is exergonic, it's going to have a little bit of energy input for it to proceed. Okay? And that's the energy of activation. All right, so reactants have to absorb a certain amount of energy to get that reaction to go, and that's the energy of activation. So we'll just symbolize it E subscript A. That's the amount of energy. The problem for cells is that there's a lot of energy of activation that would be required for every single chemical reaction that they do. And if they had to supply all of that energy of activation, they would burn up because they'd be generating too much heat. Okay? So the cell can't provide enough energy of activation to get reactions to go on their own. There's a solution, though, and the solution is to use enzymes. Okay, this is how cells get around that energy of activation problem. An enzyme is a protein, or sometimes an RNA molecule. RNAs can have enzymatic activity. There's some very important ones. All they do is they lower the energy of activation so the reaction can proceed more quickly and with less energy input. Okay. How do they do it? Well, they do a bunch of little chemical things. They may stretch bonds. If you stretch bonds, you weaken them. Okay? And that's an energy input to the system, okay, by stretching that bond. So that's an addition of energy. You may alter the reactants so they're more unstable. You can move bonds around. Okay? Move a double bond from one set of protein or one set of atoms to another set of atoms. The result is that that is a more unstable molecule. Okay? Maybe they add potential energy. They stick phosphate groups on, right? Or they use high energy electrons to achieve that energy of activation. So there's lots of different things that enzymes can do in order to lower that activation energy. Here's a sort of a diagrammatic view of the problem. This is often called the jumping bean analogy of energy of activation. There's a certain amount of energy that these reactants have to absorb in order to become unstable and to do the reaction to get the products, even if this is exergonic, and that's really what we're showing here. If we still think of potential energy level on the y-axis here, then the reactants have more energy than the products. It's exergonic. But you still have to have a certain energy input in order to get this reaction to go. Occasionally, these molecules will absorb enough energy from the environment to get over that barrier and go ahead and spontaneously react to form the products. Most of the time they don't, though. So if a cell has a reaction that it needs to have done, it needs to lower that barrier so that it's easier to go from reactants to products. It's the whole idea with energy of activation. So in the second part of this figure, an enzyme has lowered the energy of activation so that it's much easier for these molecules to absorb sufficient energy to undergo the reaction and become the products. Okay? So the reaction can proceed more easily. It'll proceed faster. Sometimes much, much faster. There are enzymes that can do their reaction 40,000 times a second, which is pretty amazing. Okay? So enzymes allow the cell to do reactions it could not otherwise do. Basically what we're doing here is looking at energy over time, and the reactants without the enzyme would have to absorb quite a lot of energy in order to proceed to the products. With the enzyme, the amount of energy, that red line, is much reduced. So it's much easier, it's much faster to do that reaction. There's still the same net change in energy, however. Okay, so how do enzymes actually work. We have an enzyme here, a protein. Okay, it's available with what's called an active site. That active site is where molecules will bind to the enzyme. Those molecules that bind to that enzyme are called substrates. Okay, the enzyme is usually very specific as to what substrates it recognizes. This enzyme, for example, recognizes sucrose and no other molecule. The next step, then, is for that substrate to actually bind in. And there's a nifty thing that happens here. When the substrate binds, it alters the shape of the enzyme so that the substrate fits very tightly into that cavity, that active site. That's called induced fit. That induced fit often is what's necessary to add the energy of activation. So this step is where that E sub A is being added. Okay? Right there. You change the shape of the protein and maybe you stretch the bonds between those two monosaccharides. That adds potential energy. Okay, so the enzyme lowers that energy of activation because of that induced fit. And now it's very easy for this reaction to proceed. Perhaps water comes in, we hydrolyze that bond between the two monosaccharides. Bingo, now we have the two separated molecules. Once that happens, the molecules are released. Okay, we now have a molecule of glucose and a molecule of fructose. So the enzyme bound the substrate tightly. That caused it to change its shape so that it maybe stretched that bond. It added energy of activation in some way. That makes it very easy to do the particular reaction, which is to break the bond between the two monosaccharides. So that happens. So now we have the products, and the products at that point are released. The enzyme spits them out. And then this enzyme is ready to do work again. Okay, we'll stop there and continue with this next time.